Welcome to Mendel and Me, where I'll cover all things genetic and whatever I feel like, honestly. Now, today's topic will be hereditary breast cancer, and this is actually the first of a series of videos on this topic. Today, I'm going to talk about the general principles of inheritance and inheritance of breast cancer. So, this is going to be a tale of peace and BRCA1. Now, hereditary breast cancer is actually pretty rare. Most breast cancers are actually sporadic, which means that they just occur randomly, isolated cases. But in some cases, in some families, you find a clustering of breast cancer cases. So there will be multiple members of this family that have breast cancer or ovarian cancer in different generations or in the same generation. And this can also be male cases of breast cancer or prostate cancer and other types of cancer, but breast cancer is sort of the most dominant one. And in these cases, the risk is actually inherited. And the gene names or the names that come up in this context is usually BRCA1 and BRCA2. And here in this video, I'm going to talk about what BRCA1 actually is and also BRCA2 and how this works, how this breast cancer risk is inherited. But I should note that only a small percentage of breast cancers are actually inherited, and the estimates are around 5 to 10%. So if you have breast cancer or someone in your family has breast cancer, this is most likely not a case of hereditary breast cancer, but it can be, so you should be aware of this possibility. Because in these cases where it is a type of hereditary breast cancer, the risk also for so far healthy members of the family is actually pretty high. So this is actually my very first YouTube video, so be nice and don't expect any fancy graphics or major editing. I'll try to remove the worst bloopers, but apart from that, this is how it's going to be. So we have the first blooper already. So basically, this is just me, my laptop, and a series of slides. Now, why did I decide to start a YouTube channel? Because as I'm starting to experiment, this is actually a lot of work. And why now? Well, winter is coming, and so is Corona, and it's going to be a very long, very dark, and very antisocial winter, and I need something to do in the evenings apart from Netflix. In addition, I recently had a rather closer brush with BRCA1 that I would have preferred, honestly. And during that time, when I tried to explain the whole thing to my family, to my friends, I realized that there is still very little awareness of what BRCA1 is, and also a lot of misunderstandings, misconceptions, how it leads to this increased breast cancer risk and how this is inherited. And so I tried to sort of channel all the things that I learned into something useful, channel this experience into something that might help other people, and try to make really some videos that explain this in a less professional and more a layman's term. There will be, of course, a lot of scientific terms, but I'll try to explain them for more that it would be better to understand. Now, if you are like most people, you will probably, if you have heard of BRCA1 at all, heard of it in the context of an article published by Angelina Jolie in 2013 in the New York Times, where she talks about the fact that she carries a faulty gene, BRCA1, as she writes it, and her doctors estimated that she had around an 87% risk of breast cancer. Her mother already died of breast cancer, and if I remember correctly, her grandmother as well. And that she also carried a 50% risk of ovarian cancer. At the time, she was still healthy, but she opted for surgeries to remove the ovaries and the breasts to sort of get rid of this risk. Now, the article is very interesting and it's important for raising the awareness, I think, but there's still little information how this actually works within inheritance and what's the problem here. So I think it will still be very useful for me to explain it a bit more in detail. Now, BRCA1, the faulty gene she's talking about, is, well, it's an abbreviation for breast cancer 1, and it's commonly known as the breast cancer gene or one of the breast cancer genes. Unfortunately, this kind of led to the misconception that BRCA1 actually causes breast cancer. Now, if you think that is the case, I have a little bit of a shock for you, because you have the BRCA1 gene. Everyone has the BRCA1 gene. And even worse, you have 2 copies of it. So, of course, that's nothing bad. It's really very important, because BRCA1 does not cause breast cancer. What causes breast cancer is having a faulty copy of it. Now, this faulty copy can be inherited, and to explain how it can be inherited, I need to give you a bit of background first on genes, on the genome, and the general laws of inheritance or heredity. Now, a gene is sort of the elementary unit of heredity, of inheritance. That can sort of be independently inherited. You may have heard of genome or DNA. Now, the genome is basically all the genes we have. All the genes we humans have make us human, that's the genome. The DNA is the molecule or the molecules in which this genome and the genes are stored. So you can sort of imagine that in the following way. The gene is basically a recipe for some dish. The genome is the recipe book that contains all the recipes that are sort of necessary to make a buffet of dishes. The DNA is basically just the paper and the letters of this book. So the physical manifestation of this recipe book. And if you sort of take a lot of recipes from this book and cook them, you get this spread of dishes, a buffet. And if you sort of balance that correctly, you get a nicely balanced buffet. And basically, this is what happens with us humans. If we take the genes from our genome and express them in a balanced way in each cell, you get us. Now, the concept of a gene has actually been around for a very long time, much longer than the understanding of how the physical manifestation of a gene in a genome actually works. The term gene was coined in 1909, but the principle, the idea of what a gene is, was basically described or discovered by an Austrian monk named Johann Gregor Mendel. You may have heard of him, who lived in the 19th century from 1822 until 1884. And his sort of productive scientific work he performed when he was a monk, or while he was a monk in Brünn, or which is now called Brno. It used to be a part of the Austrian Empire and is now in the Czech Republic. And Now, the discovery of genes, of the concept of a gene and the laws of heredity are based on experiments, breeding experiments Johann Gregor Mendel did with peas in the cloister gardens. So this brings us to the peas of this talk. And he performed his experiments in around 7 years between 1856 and 1863, And during this time, he sort of bred plants in the cloister gardens, and he cultivated around 28,000 plants, most of them peas, but also some others. And he started his experiments with sort of focusing on distinctive characteristics of pea plants that seem to be inherited independently of each other. So these were sort of the form of seeds, the color of seeds, the color of the flowers, the form of the pods and the color of the pods, and also the shape of the stems and the size of the stems. And sort of these things could be sort of, well, for each characteristic, there were different variants, so different colors for the flowers, different colors for the pods, different forms for the pods, and so on and so on. And these different characteristics could be sort of combined with each other. So you could have white flower and a yellow pod, white flower with a green pod, but red with a yellow pod, yet with a green pod, and so on and so on. And the first step of his experiments was basically to create purebred plants that were purebred with regard to one of these characteristics. So all plants in one of these purebred populations had, for instance, just white colors. They always, no matter how often you bred them with each other, they always had white colors, or they always had red colors, or always yellow pods, and so on and so on. And once he had created these purebred plants, this is of course not something that happens naturally, so he had to do artificial pollination and prevent pollination from plants he didn't want to mate. But once he had these purebred plants, he started interbreeding them, creating hybrids of these purebreds. And let's look at how this, what happened if you just look at the flower color. So he started with red flowered purebreds, white flowered purebreds, and then he crossed them with each other. Now you can try to think of what are you expecting? What would you expect? Now, most people would say that either you get sort of a mixture of red and white flowers, or you get sort of a mixture of the red and white, so maybe some sort of rose colored flowers. But what he actually saw is that every single one of the plants in the next generation of the hybrids, this is the F1 generation, was red flowered. There were only red flowers, no single white flowers in this generation. And he did this repeatedly, and there were no single white flowers. So it seemed to be that the white had completely disappeared. But when he took these flower plants from the F1 generation, so this first hybrid, and crossed them with each other, something very strange happened, or surprising happened. I can try to think what that might have been. Now, again, most of the plants had red flowers, but every so often there was a white flowered plant. And when he sort of calculated the frequency of the red flowers versus the white flowers in this generation, he saw that there was approximately 3 red ones for every white one. So ratio 3:1. Of course not, so this was sort of a bit randomly, so there was some variation, but across a large number of experiments, he got around 1 white flower for every red. And he did a lot of other experiments for the other characteristics, combined them with each other. And from this, he finally came to sort of his laws of heredity. He didn't call them this way, but today we now very often call them the laws of heredity. And it's really amazing that from these bare numbers, some statistics that he did, that he basically deduced these general and, as we know today, generally applicable laws. This was before we had computers, before biostatistics was actually a thing, or bioinformatics or anything. So now the important part of his laws of hereditary was that there were discrete units of inheritance. These were his characters, or Merkmale, he called them, since he was German. So this was the flower color, seed shape, and so on and so on. Today, we call these discrete units the genes. And for each character, there could be different forms. So for instance, the red versus the white flowers. Today, we call these the alleles, or sometimes also variants, or, but alleles is sort of the scientific term. And I mean, this is really the amazing thing, a part of his deduction, is that he deduced that each individuum had to have 2 copies or 2 alleles of each character, of each gene. And one of these alleles is inherited from the one parent, and the other is inherited from the other parent. And the reason why we see this strange distribution of colors is that one allele is actually dominant over the other. Now, let's look at the example again to really explain what that means. Now, what we see when we look at these flowers, the flower colors, is what's called a phenotype in biology. The underlying genes and alleles that determine this color is what we call the genotype. So, since we start with purebred plants at the beginning, each of the red colored plants would have 2 alleles of the red color gene variant. And each of the white colored flowers would have 2 white alleles. Now, when you breed them with each other, you will always get one of the genes or one of the alleles from the one parent and one of the alleles from the other parent. And it's pretty random which of these will be chosen. So there's a 50% chance that the one allele will be chosen, a 50% chance that the other will be chosen, and the same is the case here. But since both are either red or white, of course, every plant in the first generation, every offspring has one red and one white allele. And since we have this dominance of one allele over the other, in this case, red is dominant over white, this means that every plant will have the red color for the flower, because red is dominant and every plant in the first generation will have the allele for red. But now when you cross these plants in the first generation with each other, each of them has one red and one white. And it's always a random choice whether the red allele from the one parent is chosen or the white one. So if you choose one red from the one parent and one red from the other parent, you get a red-red child. If you choose one red from the one parent and a white from the other, you get red-white. And vice versa, if you get white from the one parent, red from the other, you get white-red. But in both cases, no matter if it's red-white or white-red, you have the red flower color. But only if you choose the white allele from the one parent and the white allele from the other parent, you get white-white and a white flower. And because this is basically random, so there's always a 50% chance of choosing the red allele from the one parent or the white allele, and the same is for the other parent, and this is basically just combinatorics. This means there is a 25% chance of having red-red, a 50% chance of having either red-white or white-red, because 25% plus 25%, and a 25% chance of getting white. And now Mendel used a very mathematic-like notation to describe the genotype. So he used letters. Big letter is for a dominant allele, a small letter is for a recessive allele. So this red would be big A, while the white would be small a, and red white would be big A, small a. And he wouldn't look at the order, so red white or white red is basically the same. So so this one here, red is again big A. We have here two big A, small a's, and small a. Now, what you should remember is that these are just, well, the probabilities, and taking over a large sample of breeding experiments, you would see that the fraction of cases, of having all of these cases, would closely resemble sort of the overall probabilities. But of course, if you just have one breeding experiment, there might be a huge variation. You could get everything from all white in the F2 generation to all red. Some of the scenarios is of course more likely. So 3 red and 1 white would be the most likely case, but basically you could get everything. Now, one example is usually used for humans to explain how this inheritance works is the eye color. So suppose most of you know that the brown eye color is dominant over the blue eye color, which is recessive. Now, if we use the notation of Mendel, this means that brown means that a brown eye color phenotype means that you have either a big A genotype, with big A being the allele for the brown color, or a big A small a, because it's dominant over small a. While the blue eye color means you have a small a genotype, sorry. Now, if two parents with a blue color and a brown eye color get a child, and kind of depends what the probabilities are for the eye color of the child. So if one of the parent has the big A genotype, then all of the children will have brown eye color, and all of them will have the big A small a genotype. Now, if the one parent with the brown eyes has actually the big A small I genotype, which we call heterozygote because it's two different types of alleles, then we go back to the 50-50 chance. So the child will definitely inherit an allele for blue eye color from the one parent, the one with blue eyes, and it might either inherit the allele for the brown eye color, the big A allele from the other parent, or the small a allele for the blue eye color from the second parent. So there's basically a 50% chance for a child with a blue eye color and a child for a brown eye color. So it's really not that surprising that if a brown-eyed person and a blue-eyed person have a child, that the child has blue eyes as well. And also, often people are saying, the child comes after the parent with blue eyes, but actually, and this is of course always the case, every child comes after both parents, because they will inherit half of their genes or half of the genome from the one parent and the other half from the other parent. And you don't get a blue eye without two blue eyes. The blue eye allele from both parents. Now, what would really be surprising is if you have two blue-eyed parents getting a brown-eyed child, then something might be off there. But again, as with all things genetic, that's a bit simplified. So as always, it's complicated because there's actually not just one gene. but at least 2 genes associated with eye color variations, and there may be as many as 16 different genes. So eye color is actually not a perfect example for this type of inheritance, which we now call Mendelian inheritance, or also monogenic, which means it's just the trait is inherited by one gene alone. But actually, there are very few Mendelian traits in human. Most traits are shaped by multiple genes, and this is also the case with genetic diseases. There are very few genetic diseases that are really just shaped by a single gene. Now let's get back to Mendel. So after this 7 years of hard work and a lot of statistics and deducing his models, he finally presented the results of his experiments and also his model, his own inheritance, first in a talk and later a paper based on a talk, which is in German, was called Versuche über Pflanzenhybriden. In English, it's Experiments on Plant Hybrids. And he presented this to the Society of Natural Scientists in Brünn, so where he lived, and later published it in their proceedings. Now, this might seem a bit obscure, but that was actually sort of what one did at that time. One presented it to one's scientific communities and published in their proceedings, because at that time scientific journals as we know them now did not really exist. So Nature, sort of one of the big journals, did not publish its first issue until 1869. Science actually was only founded in 1880. So at this time, this is what one did. And he also printed copies of his article, sent it to researchers, natural scientists around Europe or the world, maybe, I don't know. And I think Darwin even had a copy, but he didn't really read it. And unfortunately, that was a case for most of these copies. So the work of Mendel at first was largely ignored. So over the next 30 years, there are just a few citations, and he didn't really make a splash with it. He was apparently convinced until his death that one day the time of his theory would come, and it did. Now, there are many reasons why his work was ignored at first. One probably was the title. You don't really expect any groundbreaking results on inheritance in a paper on experiments on plant hybrids. And also it seemed to contradict what people were actually seeing when they looked at, well, plant hybrids, but humans, where there was more an apparent blending of many inherited traits and very few cases of what Mendel was describing, this sort of independent inheritance of these traits, or combinatorial inheritance. Now, today we know that this is actually the case because most of these traits are actually shaped by multiple genes, so they are multigenic traits. But of course, this took a while to figure out. Now, one reason which I read on a few pages was that it was also ignored because he used very abstract terminology, which was very difficult to understand. So this is just a small bit from the English translation of his paper. And you can see he used a very math-like notation. So we already counted this big A, small a, but here you have all these combinatorics and sums and so on and so on. And I think this really, really made it difficult to understand for his audience. I mean, this was before long before quantitative biology was a thing, or biomathematics, biostatistics, or bioinformatics, as we call it today. And even today, you could have many biologists run for the hills if you include a few equations in your paper. So yeah, that sounds pretty probable to me, that people really just didn't understand it and didn't make the effort to understand it. But eventually his work was rediscovered in 1900, at the beginning of the 20th century, and well, genetics never looked back. Okay, but let's get back to BRCA1. What does this whole X-course on peas have to do with BRCA1? Now, BRCA1 is a gene. So there are different alleles, different variants of this gene, and you inherit one allele from each of your parents. Now, if you're lucky and you only inherit function alleles, this means you're actually protected against cancer. So the functional alleles of the BRCA1 genes are protective against cancer. But the faulty alleles, which means there are mutations in this gene, in this allele, means that there is an increased risk for certain cancers. So in particular, breast cancer, ovarian cancer, but also slightly increased risk for other cancers. And unfortunately, faulty is the dominant, which means if you have one copy of faulty, you have the increased risk. So if you go back to the pea plants, but now imagine that the red color is the breast cancer risk, and the white color is the protective allele. Small a is the protective allele, big A is the faulty allele. And you have one parent with a faulty allele. So if it's the big A, small a genotype, you have a 50% chance of inheriting that faulty allele. Now, why didn't I put the case of the big A genotype on here? Because unfortunately, or fortunately, that depends on your view, BRCA1 is so important that the big A genotype, so 2 faulty alleles, is lethally in the embryonic stage. So yeah, well, the embryo will be aborted, is not capable of living. So you will not find any adults with 2 copies of the faulty gene. The worst case is 1 copy of the faulty gene. So this means, provided your other parent has 2 functional ones, you have a 50% chance of inheriting the breast cancer risk from your parent. Okay, so BRCA1 is not the only breast cancer gene. It was just the first one discovered, which is why it's called breast cancer 1. Shortly after that, breast cancer 2, BRCA1 was discovered, and these are the ones that are associated with the highest risk. There are a number of genes which are associated, or where faulty alleles are associated with an increased breast cancer risk, like PALB2, CHEK2, CDH1. So these are gene names. You don't need to understand what they mean. It's just different genes. But the risk is, well, it's increased to a random person in the population, but it's not, by far not as high as the BRCA1 or BRCA2 faulty alleles. Okay, so this is already a pretty long video, so I'll wrap that up here. Now, the next videos in the series I have planned, and let's see how long it takes me to make them, is the first one is on should you get tested for hereditary breast cancer, and also can you. Now, the can you question is easy to answer. Yes, you can. Now, the should you get tested question is not so easy. It depends, and we'll talk about it in the next video. But I also want to make a video about the molecular biology of hereditary breast cancer. So what happens really to lead to this increased risk. And finally, I'm planning a video on the hunt for the breast cancer gene. That's actually a pretty interesting story. Yeah, but of course, if you're just interested sort of in general principle, it's probably not important to watch that video for you. Okay, so I hope you enjoyed this video. And if you have any questions, if anything was unclear, just leave some comments below and I'll try to answer them in one of the next videos or make a completely new video for that. Thank you.